Hi Bio30! Welcome to my video where I'm going to explain what the synapse is. So, when I look at two neurons that come together, they're not actually touching. There's a very small space between the dendrites and the axon endings. So, the impulses have to jump from one neuron to the next over that space, which we call the synaptic cleft. So a synapse is the space between the two neurons. It's really small, so it can be around 20 nanometers. Really tiny. Okay, you wouldn't even be able to see this with a microscope. It's so small. It takes a little while for the chemical signal to be passed from one side of the synapse to the other. Okay, and remember that there's going to be a whole bunch of synapses between two neurons because you have a whole bunch of dendrites, a whole bunch of axon endings, so the amount of connections between them is going to be more than just one. And we use the word presynaptic and postsynaptic to describe the first neuron would be the presynaptic, before the synapse, pre as in before, and then postsynaptic neuron is the one after the space. And in order to send a signal, we use something called a neurotransmitter, which is a chemical that moves across the synapse. It's in these synaptic vesicles, which are like these little nuggets of neurotransmitters that are found within the presynaptic knob, is what we call it. They use exocytosis, which is basically when the membrane engulfs something and allows whatever is inside of it on the outside of the membrane. And then those neurotransmitters can basically diffuse across the space and attach to channels and allow them to open up. The one that we're going to be talking about is an excitatory neurotransmitter, which means that it's turning something on. There's also inhibitory, which means that it turns it off, but we're not really going to refer to that in this video because we're only talking about one neurotransmitter, which is called acetylcholine, okay, which is, again, an excitatory neurotransmitter that only attaches to sodium channels and allows sodium channels to open and allow sodium into the neuron. So just a reminder that when we look at neurons, there's a whole bunch of them that kind of connect at the end of the axons. So you might have a whole bunch of different options of where the impulse can go. So I'm going to be looking at between the axon end and the dendrite of two neurons. So the first part, the axon ending of the first neuron, we call the presynaptic knob because it's before the synapse or the space. It's filled with these things called synaptic vesicles, which are filled with neurotransmitters. We're only talking about acetylcholine for this, but there are multiple different types of neurotransmitters that you guys can have. So for example, dopamine or serotonin, those are examples of neurotransmitters. We're not going to get into each one though, just acetylcholine. And then we also have, after the little space, the dendrite of the second neuron, which we will refer to as the postsynaptic knob. The space in the middle can be called the synaptic cleft, and this whole thing is the synapse. So to get into what happens with this whole process in order to jump over this space and send the impulse to the next part, or to depolarize, send an action potential to the postsynaptic knob. That's the whole point of this, to depolarize the postsynaptic knob. The first thing that we have to do is we're going to see the nerve impulse kind of going down our axon. So that's going to be shown as sodium kind of rushing in, those sodium channels opening and going into a depolarization. Okay, when that happens, it signals another ion, calcium. Calcium is going to basically tell some of these synaptic vesicles to start to move towards the very end of the presynaptic knob. So it's going to move over and over and over and eventually get right to the end. And the next thing that's going to happen is this membrane is going to kind of engulf the vesicle and allow these neurotransmitters to be on the outside of the presynaptic knob rather than on the inside. So that's kind of what that would look like. Okay, now these acetylcholine are on the outside and can start to flow across into the synaptic cleft. So they're going to diffuse across and eventually make it to the postsynaptic knob. So I'm just going to zoom in on this postsynaptic knob because we need to see it a little bit more in depth. So once I zoom in, I notice that there's a bunch of these channels. They're sodium channels and they have little receptor sites on them. So these receptor sites are a perfect fit for acetylcholine. Once acetylcholine attaches to them, they can actually open up and allow all the sodium that might be hanging around in the fluid that basically fills the cavity around the neurons can start to go inside this postsynaptic knob and eventually depolarize the postsynaptic neuron. So we're going to see all of this acetylcholine attaching to the receptor sites, okay, allowing them to open those channels and allowing sodium in. Okay, so once the channels open, sodium will begin to flood into the postsynaptic knob. Okay, this sodium will travel down the dendrite into the cell body and through there, and then into the axon, the space between the two axon membranes that's filled with cytoplasm, and that's going to start that chain reaction of sodium channels that open up and cause it to go through that depolarization. Okay, so this is really important that you guys understand this phrase, the wave of depolarization down the postsynaptic axon. That's the whole point of this process, is to get this to happen. So we don't want the sodium channels to be open all the time. So in order for this to not have it happen all the time, we're going to allow an enzyme to come along called cholinesterase. And cholinesterase is going to basically remove all the acetylcholine from the receptors and allow these channels to close so that we don't have a constant state of depolarization, which would basically end up being like tremors if you see that from like kind of a full body perspective. If you had a bunch of neurons that were going through this constant state of depolarization, your brain would think that you're getting stimuli over and over and over again, and you would end up getting muscle tremors and things like that. So cholinesterase is going to come along, and it's going to attach to all of the acetylcholine molecules and remove them. And once that happens, those channels will close, and the sodium can stop flooding into them. So at the end, once that the channels close, basically what's going to happen is the acetylcholine will get removed from the cholinesterase, and it will actually go back to its original position in the presynaptic knob in the synaptic vesicles. So it can get used again, and this whole process can start again from the very beginning. So that is how a synapse works. Okay, you do need to understand the diagram and what's happening in the step-by-step process. So make sure you have the diagram drawn out a few times, and you should be able to tell me all of the steps from 1 to 8. So if you have any questions about this video, please come and ask me before your guys' drawing quiz. If not, I'll see you all tomorrow. Have a wonderful evening and good night.